other day, when we talked about the dog breed project, remember we did that little discussion about how the humans have always we figured out pretty early on that offspring resemble their parents, right? I gave you the example of the cows. Remember that classic example? So this kind of builds on that basic concept that traits seem to be passed down from parents to their offspring. I'm going to introduce you today to, of course, I love all scientists, but I have 3 science heroes. Albert Einstein, Charles Darwin, and this dude here, Gregor Mendel. Gregor Mendel, what I love about him is he was not actually trained as a scientist. He was a priest. And specifically, he was a monk. And who knows what a monastery is? Lawrence?
That's like a temple.
Yeah, kind of. But a monastery, and especially back in the day, he lived in the 1800s. They were these self-contained communities. So the priests, in addition to prayer and their rituals, and everybody had a job to do in the monastery. And Mendel's job was he kind of headed up the gardens because they grew all their own food. Okay. The thing about Mendel, that even though he wasn't trained as a scientist, he is one of the best scientists that ever lived because he was super, super observant. What's that word mean? Observant. Jordan?
Like he always wanted to learn new things.
That's not what that's curious, which is another piece of what he was. But
He paid very close attention to everything.
he paid very close attention to things. And so he made observations, and then the curiosity kicked in, because based off of his observations, he just had this burning need to know what's going on here. So I'm going to show you Mendel's observations first. And the plants that Mendel focused in on were pea plants, because he had some really interesting observations about the pea plants. There were 7 sets of traits that he noticed. Some had to do with the seeds, the pods, and then the plants themselves. And so in this example, he noticed that the shape of the pod, they were either all smooth or they were all pinched, and they were never in between. They were never, a little bit smooth and a little, no, that didn't happen. The seed color, it was either yellow, green, you never got a yellowish green. The height of the plant, they were either tall or they were really short. They never got kind of the in-between medium height plant. And so this is what he based his observations off from. So, and it happened over and over again. He noticed that when he took parents that had puff pods and some that had pinched, all he saw was puff. Oops. He took a yellow seed, a parent that had yellow seeds, and crossed it with another parent that had green seeds. He only saw yellow. So these were his initial observations. And this is where he started with the curiosity thing, going, what is going on here? So he made a hypothesis. He made an educated guess. He said, there must be 2 factors involved. And right here, let me just tell you. Gregor Mendel figured out what he figured out before anybody ever discovered DNA. So he didn't know about the nucleus of a cell contains this stuff that controls your traits. He didn't know this. Nobody had figured that out yet. But he said there's got to be 2 factors that are controlling these traits. And you get one from one parent and one from the other. To do what he did, he had to kind of manipulate the pea plants a little bit. Pea plants, like a lot of plants, can reproduce in two ways, because the flowers have both male and female structures. So you have the pollen makers, these are the stamens, and these produce the pollen. But then you have the ovary and the pistil, which are the female parts of the flower. So can you see that if you leave that plant alone, you might have a pollinator come around, you know, a butterfly or a bee or something, and pick up some pollen grains from this flower and take it to another one. But do you see that some of the pollen could fall right inside the same flower and self-pollinate? Well, to do his experiments, he knew he couldn't allow that to happen. Okay? He had to be sure he knew exactly what he was crossing as he bred them. So look at what he did. And I want you to keep in mind that he did not do this with a couple of plants, guys. He did this thousands and thousands and thousands of times. Think about how tedious it is. Before the flower matured, before it was able to reproduce, he removed the stamens, which I don't think he did with a butter knife like that illustration. I think he probably went in like with tweezers and actually physically removed the stamens, so that flower can no longer self-pollinate. The only way this flower is going to pollinate is with pollen from another plant. So, and he could control that. So what he would do is take pollen from a plant with known traits and introduce it to this flower, and then it would produce seeds, and he could grow those seeds up into a new generation of plants. Good with that? Okay. Think about how tedious that must have been. I hope he had people helping him, like younger priests. That would be their initiate. Oh, you're going to go help Brother Mendel in the garden, and they didn't know what they were in for. All right, so let me just kind of tell you the big findings that he had. And we're going to talk about seed color, but he saw the same thing with all the traits that he looked at. And you don't have to take notes just yet. I want you to just pay attention to what I'm talking about. So, thank you. He knew for his first experiment that he was taking, we're going to look at seed color, which is either yellow or green. So he took plants that only produced yellow seeds, and he crossed them with plants that only produced green seeds. And when we're talking genetics, when you see the X, which like in math would mean multiply, this means this is one parent being bred with this parent, or crossed. Okay? So these are the parents. And the offspring, he grew this next generation of plants, and the only seed color he saw was yellow. It was like the green had disappeared. And he's like, wait a minute, what's going on? I started with green, there's no more green. Where'd it go? Okay? Then he took two of this generation. So two plants that were the original offspring. So we'll call this the first set of parents. And this is the second set of parents. So he did a yellow crossed with a yellow. Okay? Predictions? Something really curious happened. For every 3 yellow, he saw 1 green. The green came back. And he didn't just grow 4 plants. He grew thousands. So this is a ratio, okay? This is a ratio that if he grew 4,000 plants, around 3,000 of them would have had yellow seeds, and about 1,000 of them would have had green seeds, more or less. Okay? Are there any questions on done that yet. Good so far? And so this just kind of confirmed his hypothesis. He's like, okay, so there are two factors involved, and one of them seems to be stronger than the other one. Now, for those of you that have done the what is a trait, does anyone remember the term for the trait that covers up the other one? Landon? Dominant. So Mendel said these factors, remember, we didn't know the word gene at that point. We didn't know about DNA. So Mendel said these factors, one must cover up the other one. One is dominant. Okay, yep.
And the other one, do you want me to say the other one?
Yep.
Receptive?
Not receptive. Close. Recessive.
Oh.
Okay? A dominant trait, and this now should be going into your notes. Let's say dominant trait. I'm going to say covers up. And notice that I'm putting that in quotes. Covers up a recessive trait. So the recessive trait gets covered up. You're going to notice that a lot of our vocabulary, it's going to come in pairs. So our first set of terms, dominant and recessive. Are we good? No. Hurry. Type faster. All right, moving on. I'll leave that up there for a second if you're still writing. We're going to talk about the notation that we use. And if you've done what is a trait, the example that they gave you in that one was the hitchhiker's thumb versus the straight line. All right. So, the way that we notate which trait is the dominant is we use the uppercase letter. The recessive, we would notate with the lowercase letter. All right. Now, what I'm going to show you right now, I don't want you to take notes on. So if you're typing on an iPad, bottoms up, I really need you to pay attention to this, and then I'm actually going to give you a practice sheet that is what I'm going to put on the board right now. Okay? A number of years later, this dude here, his name is Reginald Punnett, was looking at Mendel's work, and he was coming at it from more of a math standpoint. He's like, you can predict the probability of what the traits in an offspring will be if you use a very systematic way of looking at it. So we're going to look at seed color. Remember, Mendel's calling them factors. We'll do a little bit more vocab in a second where we'll give that a different name. Seed color factors, Mendel knew that one of them was dominant and one of them was recessive. So we give the dominant trait gets a capital letter. So this would be the factor that would be yellow, and this would be green. Okay? Good so far? Notice that we don't use Y and G. We pick one letter when we're doing these, when we use the uppercase and the lowercase. So it's really important that when you're writing these, that you can tell your uppercase from your lowercase. Notice that I'm putting the little curly Q tail on the Y here, so there's no question what it is. You've got to be careful to do that, otherwise you're going to get confused. All right, so I'm going to show you what are called Punnett squares. And these are just a tool that can show you what's the probability that you'll see certain traits in the offspring. Can I erase this? So let's go back to Mendel's first cross. He had what he knew were pure strain plants. So he said, they must have, their pair of factors that they have must both be that. So he had a pure strain yellow that he crossed with a pure strain green. Okay? A Punnett square starts off looking a lot like a tic-tac-toe board. And then we color in that square. Okay? Do you remember our animation of meiosis? So you remember when meiosis is happening, you end up with the 4 daughter cells, and they only have half of the DNA, right? You're only getting 1 copy of each gene or each chromosome from your parents. So we're going to call, this is going to be parent 1, and this is parent 2. They're plants, so it doesn't seem appropriate to call them mom and dad. So we'll call them parent 1 and parent 2, okay? So when you think meiosis happens and we end up with the daughter cells, so what could parent 1, what could end up in their daughter cells? I could either get that one, and I'm going to put it right there, or I could get that one. What can I get from parent 2? I could get that one, or I could get that one. So in a Punnett square, along the top and the left-hand side, that's where you put the individual factors that you could get from each parent. And then these squares in here would be the offspring. So offspring are going to end up with 2 copies, right? So what's this one going to get? It's going to get this one and this one. You go over and down, over and down. This one, this one, this one, and so on down the line. Okay? So here's my question to you. What seed color do these offspring have? Raise hands. Lydia?
Yellow.
How do you know?
Because the capital Y is dominant.
They have a blend of factors. They each have one that's for the yellow and one that's the green, but we know that the yellow is dominant, and it's going to cover that up, which means that these are going to look yellow. Even though there's this factor here for green, it's going to look yellow. Does that make sense? Are there any questions on that? Isn't this what Mendel saw? It was like the green trait disappeared, right? All yellow. Good so far? All right. What did Mendel do next? He took 2 of these. Okay? So he took these offspring. So parent 1 is this, and so is parent 2. 2. Okay? So what can I get from parent 1? I could get, say it with me, say big Y, little y. Here? Okay. So now we're going to talk about the offspring. Talk me through it. What have I got?
Big one, big one.
So that's the little one.
Big Y, big Y. No, just give me the big Y, little y, okay? What's this one get?
Big Y, little y.
Big Y, little y.
Little y.
Little Y.
And here's, and this is, you know how in math they make you write things in a specific way regardless of where it is? Scientists are the same way. We write the capital letter first if there's two different ones. So this one, we'd also say big Y, little y. But what are we going to get here?
Little y, little y.
Ooh, alrighty then. So this looks different than that. What is this? What seed color?
Yellow,
Yellow?
yellow, yellow,
Green. Because the only way we can see the green trait is if they have two recessives. Because if there's a dominant there, it's going to look like the dominant trait. So in order for the recessive trait to ever show up, you need two copies of the recessive. All right? So Mendel had a 3-to-1 ratio. What have we got? 3 yellow, 1 green. Looks like 3-to-1 to me, right? So Punnett developed these tools really to just kind of work with the data that Mendel already had, and other scientists along the way. So does this kind of sort of make sense? Great. So now we're going to attach some more hefty vocabulary to all of this. Can I erase this one?
Yes.
Like I said, you're going to have your study sheet, your practice sheet, where you're going to redo this. I'm going to leave this one, because we're going to refer to it. All right. So let's do some definitions, and they do come in pairs. You already have dominant and recessive, right? All right. The next set of pairs that I want to talk about is gene. We've heard the word gene. And the definition of a gene is it's just the section of your DNA that controls a specific trait. So the example that we're talking about here is, we would say it's the gene for seed color. You writing this down, Adam? The term that goes with this is allele. An allele is a different form that the gene could have. I'm going to say, let's say the different forms, make it 12, the different forms a gene can have. So our examples from this would be, we have Does that make sense? So the gene is the actual where on your DNA is that trait controlled, and the alleles are what are the different forms that the gene could have. So if we were talking about like eye color, there could be a brown-eyed allele and a blue-eyed allele. With Mendel's pea plants, there would be one for tall, one for short. In this example, yellow seeds and green seeds. Okay.
Does the yellow seed always have to be the dominant one?
Yellow is the dominant allele. So it is, it's always, if that is present, if that allele is present, it's always going to look yellow. Because the dominant will always cover up the recessive. Other questions? Matt, shows up front. Yep.
Why couldn't the green be a dominant? Like,
It just isn't. I mean, it's just how it is. Okay, that one is, I guess, stronger than the other, and it's not a really good word to use, but it just is the one that gets covered up. I'll give you a little bit of a preview of things to come. It doesn't usually work like this. Pea plants, Mendel picked the perfect model to study because they do behave like this. So he was able to figure things out. But inheritance of like human traits is so much more complex than pea plants. And it doesn't usually work like this. There are a few that do. There are a few that do. And we're going to look at those, things like blood type and color blindness and stuff like that. But so good with this? All right, the next set of words that we're going to do, gene and allele is pretty tame. Wait till you see these next ones. The next pair is phenotype and genotype. As usual, guys, I will always give you spellings for these. Like you'd always have a word bank, or it would always be part of what is in front of you. So worry less about memorizing spelling and worry more about knowing what does that really mean. All right? So the phenotype is what the trait actually looks like. So in our example, what could the seed color be? Yellow or green. So those are your two possible phenotypes. The genotype is what alleles are actually present for that trait. So in our example right here, there are 3 different genotypes. I've got this guy here that's got big Y, big Y. I've got these 2 that have big Y, little y. And then I've got the 2 little y's. So in seed color, there are 3 possible genotypes. The traits, the phenotypes, are yellow or green, but there's 3 possible genotypes. Questions? People are still writing, so I'm giving you a minute. And your last set of 50-cent terms is heterozygous and homozygous. Heterozygous means the alleles are different from each other. What's our example here of the one that has the alleles that are different from each other? Big Y, little y. Homozygous means the alleles are the same as each other. Do you see that I have 2 possibilities here? What are my 2 possibilities here? If I look here, which ones, Jordan?
YY and YY.
They're all Y's, Jordan. You have to be more specific.
Well, the capital.
Capital, the 2 uppercase,
Caps and no caps.
or the 2 lowercase. 2 dominants and 2 recessives. So if an individual is heterozygous, it means when you look at their genotype, they have one dominant allele and one recessive. Okay, big Y, little y. If an individual is homozygous, it means that they've either got two dominants or two recessives. So if I was just talking about it, if I said that individual is heterozygous, it can only be one thing. It's got to be the combo. But if I said it's homozygous, how do you know which one? Is it this or is it this? What word should we add to that so we can make sure we're talking about the same thing? Vanessa?
Like dominant or like recessive?
Yeah. So I would call this guy here homozygous dominant. This one, homozygous recessive. You have, when you talk about homozygous, you've got to specify which one it is that you're talking about. And I'm sorry that that got a little bit low on the board. Does anyone need me to repeat anything? So we good? I'm going to leave this up here. This really needs to be in your notes. But what I'm going to give you is a practice sheet. And guys, it's exactly what the front of this is exactly what I just put on the board. Okay? How about we just focus forward right now? You're just going to recreate what I already did. But it's good to practice how to set these up. So the front is what I just put on the board. The back is a different problem to think about. And what you're doing on the back is there are two possible genotypes that look yellow, right? There's only one genotype that's ever going to look green. I want you to do one of each. You're going to cross a yellow and a green, but you're going to use the one form of yellow here and the other form there. I bet for a lot of people that's super confusing what I just said. I get it. Don't stress about this. This is going to, if you don't finish it, it's going to be homework. And I will have a line out the door during tutorial, which is absolutely fine. Come see me, work on this in tutorial if you need to. Here's what's going to happen though, is I'm going to hand this out and some of you are going to be like, boom, got it. If you think you've got it, let me check it. And I might deputize you, meaning I'm going to say, okay, could you go around and help other students who need some extra help, okay? So, any questions on what you need to do right now? All right. If you need help, just raise your hand.